Hello, welcome to 13 Universal Learning Beyond Barriers, and this is A-level CIE biology questions, and then mitotic cell cycle series 2. So if you have not watched series 1, watch it. Okay, very insightful, educative. We crack some concepts. Okay, and then we had a lot of knowledge on how to solve questions. Okay, remember this question is just to equip you with how to apply the concept you've learned. So, and this is schedule. Okay, we're going to the first question. It says immediately after which stage in mitosis in an animal cell does the cytoplasm start to divide? All right, which stage is that? So I want you to help me find out which stage. Okay, so which are the stages? Is it anaphase or metaphase or prophase or telophase? Which stage? Now if you check the word they use here, look at the word they use here. The word here, all right, start to divide. Cytoplasm start to divide. So which stage does it start to divide? Just a minute. Okay, which stage does it start to divide? All right, um which stage does it start to divide? So I want you to pause the video and try the question. Okay, so try the question. So sorry for the distraction. All right, now look at it. Cytoplasm starts to divide. Now this is synonymous to cell division. All right, so when cytoplasm starts dividing, that cell division, we call it cytoplasmic constriction. All right, and if you check, if you are conversant with cell cycle, guys, this is cell cycle. Pardon my diagram. Okay, and then we have this segment, right? This is different segments, right? We have these are all parts of cell cycle. Okay, of course this is mitosis. Mitosis is nuclear division. C is cytokinesis, which is cell division. Okay, and this is G1, is part the first interphase phase where there is growth. The cell becomes bigger and it makes protein, you know, through transcription, translation. Of course, these are going on there. New organelles are formed. Of course, this is S phase. S phase, DNA is semi-conservatively replicated. In G2, this is G2, central replicate checkpoint to detect genetic errors. Of course, in G2 we have of course transcription translation happening. All right, but look at this, guys. In prophase, in mitosis, we have about 4 stages: prophase, metaphase, to call it PMAT, anaphase, okay, and telophase. Okay, yeah. So look at it there. Do you see this? So the question says immediately after which stage in mitosis. So this is mitosis, guys, and this is the first stage. They are in sequence. Prophase occurs before metaphase, then anaphase, and then telophase. So after telophase, the next is cytokinesis, which is when the cytoplasm starts to divide. So you know the answer now. So immediately after which stage in mitosis in an animal cell does the cytoplasm start to divide? Of course, it's telophase, right? It's telophase. After telophase, we go to cytokinesis, right? That's when the cell starts to divide. All right, let's go to the next question. All right, it says the diagram shows a cell of an organism formed by reduction division. The reduction division, that's it. Reduction division is halving the chromosome number. So that means that this is haploid. Okay, so they're not asking what is the diploid number of this organism. Now reduction division is meiosis. It halves chromosome number to produce haploid cells. So this cell is haploid. The cell is haploid. Okay, so they are asking us which one is the diploid number. Okay, guys, you know that. Of course, you know that. Having done the topic, you know that diploid is 2n. Okay, and then haploid is n. Okay, 2N is diploid, two sets of chromosome. Haploid is just a set of chromosome, guys. Okay, let's check if this is produced by reduction division. That means what it has here is haploid. Let's count it. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. Okay, I think it should be 20. Yeah, let's go again. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20. So 20. So the haploid is 20. So diploid is 2 times haploid. Okay, 2 times 20 is what? Okay, 2 times 20 is what? So what do you think is the answer? Okay, that's 40. Of course, you're right. Okay, 40 is the answer. You can pause the video and look at it again. Okay, we need to hear your comments. Check our videos, make comment, like them, subscribe. All right, let's go to the next question. It says, which diagram represents a cell undergoing metaphase of mitosis? Which of them? Okay, I want you to try this. Pause this video and try it. Now this is which diagram represents a cell undergoing metaphase of mitosis? Metaphase of what? Mitosis. Okay, look at this. This A, this is prophase. How do I know? The chromosomes appear as, all right, sister chromatids joined by centromere. So our spindles are yet to start forming anywhere. This is prophase. Now look at B. B is anaphase, because sister anaphase of mitosis. The sister chromatids are being pulled apart to the opposite pole by the spindle. Spindle shorten and contract and pull sister chromatids apart, all right, to the opposite poles with centromere leading. Centromere is leading. Okay, so this is not metaphase. Now let's go to C. Okay, now one of the characteristic features of the metaphase, one of them is that the chromosomes align themselves along the equator or the metaphase plate. Okay, now these chromosomes are aligning themselves at the equator. Here they also align themselves at the equator. So, but both of them are metaphase, but this is the metaphase of meiosis because the chromosomes are paired. This is metaphase 1 of meiosis because you have pair of chromosome, bivalent chromosome, but there's no pairing here. So this is metaphase of mitosis. Okay, just have only homologous chromosomes, they're not pairing. All right, so the answer is what? C. Let's go to the next question. Caucasian, don't know the name, but pronounce it. Caucasian, whatever it is. Caucasian is a substance which inhibits the formation of spindle fibers. Now, what a minute. What forms the spindle, guys? Talk to me. What forms the spindle? Okay, the central, okay, make microtubules which are assembled to form spindles. At which stage is this cell cycle interrupted? Okay, so at which stage is spindle formed? Cell division, DNA replication, interphase, mitosis. Okay, look at it. Cell division is cytokinesis. Spindles are not formed here, right? Okay, DNA replication is in interphase. S phase of interphase. Spindles are not formed here either, if you are conversant with the features. Now interphase, of course, no spindle. G1, G2 spindles are not formed, but mitosis spindles are formed, right? Starting from the late prophase where spindles start forming. So this will be inhibited, all right, because look, which inhibits the formation of spindle. At which stage is cell interrupted? It's interrupted in mitosis because that is where spindle fibers are formed. So that is where the cell will be inhibited, all right? So let's go. So the answer is D. Let's go to the next question. It says, which statement describes a cell belonging to a haploid organism? Now, haploid is one set of chromosomes, so one set of gene that is capable. Now, it's capable of reproduction. Now, number A says, option A says it has chromosomes that contain one polynucleotide. Pause the video and solve this question. Please solve this. Fry this fish. Okay, this is your own fish. Fry it. Let me see what it got. Okay, pause it and do it. Okay, let's go. Now it says A. There's a which statement describes a cell belonging to a haploid. organism that is capable of reproduction. A says it has chromosomes that contain one polynucleotide chain. Now, no chromosomes contains one polynucleotide chain. Okay. This is talking about DNA. DNA has two strands of polynucleotide, not a strand. Okay. DNA has two strands, double-stranded, forms double helix, not one strand. This is eliminated. B says it is capable of carrying out a reduction division to form gamete. It cannot, all right? It's only diploid cells that undergo reduction division, so that at fertilization, the diploid number is actually restored, and that prevents doubling of chromosome number in each generation. So this is not possible for haploid cell. This is eliminated. C says it possesses two copies of each gene. No, it does not possess each two copies of each gene. It is haploid. It's just one set. Haploid means one set. One copy of each chromosome has one gene. It doesn't have two. Okay, that's why she's wrong. D says it will undergo cell division by mitosis. Yes, during asexual reproduction. Yeah, it can copy itself. Okay, by mitosis. See what it's trying to say, guys. Now this is this haploid. Now, pardon my drawing. Haploid is N. Okay, so yeah, this can undergo this. Okay, to give us this is mitosis. It's from two daughter cells that have the same chromosome number. Do you get? Right, two daughter cells. Right. How does this work? Of course, don't forget that this will first of all at interphase it will be doubled to give us 2n. Right. So then of course it will now undergo, right, now it will double to give us 2n, and then of course it will now give us at the end of the division we have the daughter cells having the same number of chromosomes as the parent. So this is mitosis because this is the parent. The parent cell, mitosis produces genetically identical daughter cells, right? These two cells genetically identical, they have the same chromosome number as the parents, they have the same genes. Okay, so the answer is D. All right, quickly let's go to the next question. So some chemicals used to stop tumor growth, all right, work by preventing DNA double helix from uncoiling and separating. All right, when does this happen? Tell me. In which stage of cell cycle does DNA double helix uncoil and separate? Okay, is that not synthesis phase? That's interphase, right? During which stage of the cell cycle would they act? All right, anaphase is in mitosis. Interphase, yes. Metaphase in mitosis, prophase. Now, A is anaphase, it occurs in mitosis. DNA does not uncoil in mitosis. So all of these options that have to do with mitosis, anaphase, metaphase, prophase, they are wrong. Now, interphase is correct. Why? Because in the S phase of interphase, DNA undergoes semi-conservative replication, and that is when DNA uncoils, separate. Okay, do you remember? Okay, this double helix. Okay, double helix. It separates and uncoils so that each of them can be used as a template. Right. All right. Now sometimes forms a replication file. Okay. All right. So the answer is in interphase. Right. So that is interphase. Let's quickly. Noko. Pronounce it. What? Nocodazole. Wow, what a name. Is a chemical used in the study of mitosis. Good. It causes all mitotic cells to stop dividing at metaphase. So it makes them to stop dividing at metaphase. Which statements correctly identify how this chemical might work? So they stop dividing at metaphase. Number one says inhibits chromatin condensation in the nucleus. Does this happen at metaphase? Okay, pause this video. Look, pause it. It's time for a turn. Tell me the answer now, and I'll buy a chocolate. Okay, if you get the answer, I'll buy a chocolate. I mean it, seriously. Now look at it. One says inhibits chromatin condensing in nucleus. This occurs, chromatin condensation occurs in prophase. Okay, not in metaphase. Two says prevent replication of the central. Central replicates in G1 sorry, G2 of interphase. That's where central replicates, and that's not metaphase. There is a stop sister chromatid migrating to opposite poles. Okay, yes, because after metaphase, the next is this. All right, so from at metaphase they line up, and but the next one is anaphase, and they're going to migrate. So if you stop it at metaphase, that means the next phase, which is anaphase, where sister chromatids migrate to opposite poles, will not occur. This will be inhibited. All right, so 3 only. Okay, chromosome telomeres are essential for DNA replication and are not completely replaced during mitosis. A substance X is known that completely replaces telomeres during mitosis. What will be the effect of growing cells with and without substance X? All right, so I'd like you to try this. So telomeres, what are telomeres? Telomeres are repetitive base sequences that are found at the tip of chromosomes. They protect genes. They prevent loss of genes or loss of genetic information. Okay, and then of course they determine the lifespan of a cell, because the length of telomeres determines how long the cell is going to divide. Once telomere is completely depleted, the cell will die because it will start losing some genes, and once it loses genes, it may not be able to survive because it won't be able to make some important proteins. So telomeres are at the tip here. Okay, all right, now is at the tip, this tip here, protect genes. All right, so remember, telomeres, this substance X, all right, replaces it. Then this, I guess this substance X is maybe telomerase enzyme that replenishes it. Okay, they say what will happen? So with substance X, yeah, if you replenish telomeres, all right, that means the cells continues to divide. So which option? Cells divide more, not more rapidly, but it continues to divide. Cell division doesn't slow down. Okay, because as long as we have telomeres, cells will continue. So telomeres allow DNA replication or cells to continue to replicate or divide. Okay, so that is why if we have, you see, in normal cells, the telomerase enzymes, which I think is X, is inactive. All right, but in cancer cells and stem cells, is very active. That's why cancer cells and stem cells divide continuously, right? So so because A seems to be, of course, the answer here, when you remove it, cells division eventually slows and stops, and the cell dies. This is the reason why cells die. This is called apoptosis. It's called programmed cell death. Every cell dies because once telomere is depleted, genes are lost and cells will die. Okay, this is the reason for aging, and once this is fixed, human beings will not age. So scientists are looking for a way to activate telomerase enzymes that will replenish this. All right, so that we don't age again. Imagine a world where people don't age; they remain young forever, forever young. Okay, so the answer is A. Good. Let's go to the next question. Time is gone. The diagram shows a cell nucleus in prophase of mitosis. So that's prophase. Which statement describes the chromosomes found in each daughter nucleus immediately following division of this cell by mitosis? Oh, so look, please remember to subscribe to our videos, like it. Remember also to make comments and share. Now, immediately after you see, the question says, which statement describes the chromosomes found in each daughter nucleus immediately following division of this cell by mitosis? Of course, each of them, see, each of them, their sister chromatids will separate. So this will go, this will separate, this will separate, this will separate. But you see that these chromatids are actually, they are genetically identical because they have the same genes. Okay, all right. And then let's look at this. A says we're going to have 4 chromosomes. Yes, this is it. This is going to be chromosome 1, chromosome 2. Each chromatid will represent a chromosome. Okay, so when we separate them, each cell, daughter cell, will have something like this. Okay, see what I'm trying to say. Okay, a daughter cell will have just one daughter cell. I just want to show you one daughter cell here. Okay, pardon me. Okay, it will have, okay, these chromatids, 8 of them. 8 of them. All right. 8 of them. Let me not continue. All right, yes, it's going to have 8, and each, okay, yes, let's go back. 8 chromosomes, each consists of 4 chromatids. No, each will have 1 chromatid. All right, 1 chromatid represents 1 chromosome, right? We split it. B says 8 chromosomes each contain 1 molecule of DNA. Yes, because one chromatid contains 1 DNA molecule and 8 histone proteins. Okay, so B makes a lot of sense. C says 4 chromosomes each consisting of 4 chromatids. No, we're going to have, all right, 4 chromatid, 8 chromatids. Okay, each represents a particular chromosome. D says 16 chromosome. No. It's not going to double the chromosome. It's not going to double. It's not halving the chromosome. C and D are outright wrong. This is not halving it. This is not meiosis. All right. Neither this is doubling it. Okay. All right. So the answer is B. 8 chromosomes. Okay. Chromatids. Each contains 1 molecule of DNA. All right. So I want to hear your comments. Let's hear what you're thinking. All right. Which features of mitosis ensures that genetic constitution of the cell is maintained? Yeah. Pause the video and look at it. What is your answer? Now, number 1 says the position of the chromosomes on the equator. The position of the chromosomes on the equator of the spindle. Now, the position has nothing to do with maintenance of genetic information. This says the longitudinal division of centromere. Yeah, I think this is very important. What do you think? The centromere has to split, right? You know centromere? Centromere is that structure that connects 2 sister chromatids. It joins them together. It also attaches them to the spindle, right? So you see, it has to split, because if it does not split, they will not move apart. These 2 sister chromatids, they have the same genes, so they have to move apart, right? So it has to divide longitudinally so they can move apart to the opposite pole, making sure that each daughter cell contains one of it is, okay, because they carry the same genes, and that makes sure that they are genetically identical. 3 says the DNA of the parent cells replicate before mitosis. Yes, the DNA replicates. Semi-conservative replication of DNA occurs, making sure that the exact genes, all right, in the parent DNA molecule is copied in the daughter DNA molecules. So 3 is correct. 4 says pulling apart of chromatids to opposite poles. Yes, this happens at anaphase. 2 of them must be pulled apart to the opposite poles. Okay, so that each pole will form a new daughter cells, and because they are pulled apart, each daughter cell will have exactly the same chromatids like the other one. So 2, 3, 4. Get the answer. All right. So guys, this was the thank you very much for listening to this video. This is Shedro, and we're going to be posting later, doing series 3. Please subscribe to our channels, like it, share it, stay with us. We have lots of videos for you. Have a wonderful time. Thank you. Bye.